So in today's lesson, we will be looking at anaerobic respiration. So the learning outcome for this lesson will be 12.2.10, outline respiration in anaerobic conditions. That means when there is no oxygen, okay? No oxygen. So for mammals, it will go into lactate fermentation. And then for yeast cells, it will go into ethanol fermentation. So both of these are under anaerobic respiration. So for mammals, it will produce lactate. So that's why it's called lactate fermentation. For yeast cells, it will produce ethanol. So it is called ethanol fermentation. So fermentation, the definition is actually the oxidative, sorry, it's not oxidative, it's the breakdown of organic molecules, okay, by organisms to produce different types of products, okay. So that's the definition of fermentation from Biology Dictionary. It's a breakdown of organic molecules by organism to produce different kind of products, and usually in a during the stress condition, in a stress condition. So in this case here, the stress here is lack of oxygen. It could be other stresses, okay? So therefore, I will say that anaerobic respiration is actually a subset of fermentation, because fermentation is not only about anaerobic respiration, it also includes a lot of other different processes that break down organic molecules to produce different kinds of products, okay? Right. So next learning outcome is explain why the energy yield from respiration in aerobic conditions is actually much greater than in anaerobic conditions. So take note here that a detailed account, that means counter of the total yield of ATP from the aerobic respiration of glucose is not expected. Like what I mentioned, it's not in syllabus, but I need to show you the account of it so that eventually we can tell why is it 38 ATP from aerobic respiration. Why is it only 2 ATP from anaerobic respiration? Okay. So in the previous lesson, we actually looked at oxidative phosphorylation. In other words, it's called respiratory chain. So let us look at the entire processes, because this process is important for us to understand anaerobic respiration. Okay. So in oxidative phosphorylation, we recap. First step is oxidation of NADH and FADH, where they will release the 2 hydrogen atoms carried by them, each of them, okay? Then hydrogen atoms will be transferred by the hydrogen carriers. Then it will split into H+ and electrons. And then the electrons will be transferred by the series of electron carriers in ETC. ETC stands for electron transport chain. Then oxygen will act as a terminal proton and electron acceptor to form water. Then proton gradient will be generated from the electron transfer in ETC. So when electron is being transferred in ETC, proton are being pumped, okay, from the matrix to intermembrane space, generating proton gradient. So chemiosmosis will happen. That means when the proton diffuse across a stock particle, which consists of proton channel protein and ATP synthase, ATP is synthesized from ADP and PI. Okay, so then we also look at the ATP count. Theoretically, is 38 ATP per glucose molecule fully oxidized. Practically, is 32 ATP per glucose molecule fully oxidized after minusing the ATP that will be used by the mitochondrion itself to pump ADP into the mitochondrion and pump ATP out of mitochondrion, okay? So now we're going to look at what happens to respiratory pathway when there's no oxygen. So if you want to understand this, we must always start from the process when oxygen is directly involved. So when oxygen is not available, then the reaction will not occur, okay? So we're going to look at it step by step backwards, starting from the reaction which needs oxygen, linking it to the rest of the reaction that's linked to this one. So this is a chart that you are familiar with, which actually shows the oxidative phosphorylation. Okay, so we know exactly this is where oxygen will be required in the process. So it will accept the 2H plus and 2 electrons to form water. So therefore, when oxygen is not present, that means to say this reaction will not be able to occur because oxygen is a substrate. So no substrate, no product. So no reaction. So when there's no oxygen, no final or terminal proton electron acceptor, water will not be formed. And other than that, you know that, so here I already removed water. You can see water removed already. Huh? So then, because there's no oxygen to accept the electrons that are being carried by this electron carrier, 2e, it will not be able to be passed to the oxygen because no oxygen. So electron carrier, this electron carrier continues to carry these 2 electrons. No? So which means to say this electron carrier is already saturated with the electron, just like enzyme saturated with substrate, the carrier saturated with the electron that it carries. So because it's saturated, it is unable to accept the 2 electrons from its neighbor. So there are 2 electrons here. These 2 electrons cannot be passed to this electron carrier because this electron carrier is saturated, so it cannot be passed. So in the same way, these 2 electrons also cannot be passed to this electron carrier because this electron carrier is also saturated. So which means to say, in the entire ETC here, so this whole thing is the entire ETC here, we can say that there is no electron being transferred in this ETC. So when electron is not being transferred, I'm going to call you already, so you need to help me to complete the story. So when electron is not being transferred by ETC, what is the next reaction that's directly linked to this that cannot occur? Filbert. What is the next reaction that's linked to this reaction that when this reaction stops, that reaction must stop? Filbert? So correct, you say that because this electron carrier is saturated, so this hydrogen carrier that carries this hydrogen, right, when it will not split to proton and electron because there's no carrier to receive this electron. So these 2 hydrogen will remain inside the hydrogen carrier, which is saturated now. So another 2 hydrogen here, 2 hydrogen atoms here, okay, will also unable to be transferred to the next hydrogen carrier because it's already saturated. So because of that, eventually these 2 hydrogen, okay, one hydrogen atom, the other one is hydrogen ion, both cannot be released into this hydrogen carrier. So this is what you say, right? And this hydrogen here from FAD and the H plus, this one also cannot be released to this hydrogen carrier. So cannot be released, cannot be released. So that means to say that this reaction, oxidation of NADH and FADH cannot happen. So it cannot happen, that means the product NAD plus and the FAD plus will not be, can you give me the term? Will not be, what is the name of the process? Chujiaying?
Regeneration.
Very good. It will not be regenerated. So whenever you want to say regenerate, means if it is something that's important for the cell, that's why it has to regenerate it, right? So it cannot be regenerated. So NAD plus and FAD plus cannot be regenerated. Very good. So up to here first, up to here. Now we want to look at the entire process of hydrogen transfer and electron transfer, right? What is so important about the electrons must be able to be transferred in ETC? What will it lead to if it's that's being transferred? Januli?
The energy will be released, which can be used to pump the hydrogen ions to the intermembrane space.
So that means to say whenever electrons are being transferred along ETC and the 2 hydrogen atoms transferred through the hydrogen carriers, then the proton can be pumped, right? There'll be excess energy released and proton can be pumped Pump means it always goes against concentration gradient, right? So from a region of lower to higher concentration, right? So therefore, electron can be pumped when there's electron flow, proton can be pumped when there's electron flow. So there'll be proton gradient. And because of the proton gradient, proton can diffuse across a stock particle through the proton channel protein and ATP synthase, so ATP can be synthesized. So now when there's no oxygen, there's no electron being transferred along ETC, there's no hydrogen atoms being transferred along the hydrogen carriers. So that means to say there'll be no proton being pumped. So therefore I can write here, no proton being pumped. This process cannot happen, this process can happen, this process can happen. So there'll be no proton gradient. Proton cannot diffuse across stock particle. No ATP synthesized. So this reaction will stop. This reaction will stop. All these reactions here will stop. Correct? So can you see how you can link from when there's no oxygen immediately? This is the reaction that involves oxygen. So what is the next reaction before it? Before the oxygen is this one. Okay, let me delete the whole thing. So whenever there's no oxygen, that means this step that's involved, directly involving oxygen will not happen. So the step before it is this one cannot happen. So this one cannot happen, this one can happen, this one can happen, this one can happen. Then if this one can happen, that means this can happen, this can happen, this cannot happen, this can happen. Then this can happen, this can happen, this can happen. Okay, and this process also can happen, this process can happen. So this one cannot be produced, this one cannot be produced. Okay, so it's very easy. As long as you learn up oxidative phosphorylation, you'll be able to actually explain what happens when there's no oxygen. So our main concern here is, okay, not only I cannot produce ATP, and there's another two important molecules that I cannot produce. These two are very, very important, apart from ATP. These two are very important. Okay, why are they important? Later on you see. Here, without NAD plus and FAD plus, all the dehydrogenation reactions in the Krebs cycle and the link reaction cannot occur. Now, you already know by now that every time we want to oxidize the organic molecule by respiration, for example, C6H12O6, right? So in the respiration is always about removal of carbon as well as removal of hydrogen, right? We already said that carbon is removed by decarboxylation to produce CO2. And then H2, hydrogen is removed and it must be carried by these two carriers. Okay, either this carrier or this carrier. It must be carried by these two carriers to be brought to the inner membrane of mitochondria to undergo oxidative phosphorylation. Then only it can be used to produce the ATP that we want. Because oxygen is used to oxidize them, so ATP can be synthesized. Okay, so the NAD plus and FAD plus are very, very important in all the dehydrogenation reactions in Krebs cycle and link reaction in order to carry this hydrogen to be oxidized to produce ATP. So which means to say, when there's no oxygen, NAD plus and FAD plus cannot be regenerated. So not regenerated. So which means to say my Krebs cycle and my link reaction will have to stop. So let's just look at the Krebs cycle and link reaction. So this is a Krebs cycle here. So you know that in Krebs cycle, right, where are the regions that need NAD and FAD plus? So from citrate to alpha ketoglutarate, there's one decarboxylation and one dehydrogenation. So this one needs NAD plus. From alpha ketoglutarate to oxaloacetate, you need to memorize there are 2 NAD plus needed and 1 FAD plus needed. Okay, so that's where whenever no NAD plus, no NAD plus, and no FAD plus, the entire Krebs cycle cannot occur. Same as for link reaction. So this is where the NAD plus and FAD plus are needed. Same for link reaction here. So link reaction involve one decarboxylation and one dehydrogenation reaction. So when you don't have NAD plus, of course, this link reaction will not occur. Okay. So what about glycolysis? Can glycolysis occur or not? Theoretically, it must not be able to occur because NAD plus is not generated, not regenerated. But glycolysis, whenever there's no oxygen, it can go into the anaerobic pathway in order to regenerate back the NAD plus. Okay, so the anaerobic pathway is to regenerate back the NAD plus so that at least glycolysis can happen. Why you want glycolysis to happen? Because now there's no oxygen, ATP cannot be synthesized. But you still have to run. Let's say, for example, you exercise halfway or you encounter a danger. You need to run away from the danger. So your muscle have to continuously contract one. So you must keep on supplying ATP to your muscle, whether you got oxygen or not. Your muscle need to contract. So therefore, our cell will go into to see, okay, now I'm shortage of oxygen, but I still need ATP. So what can I do now? So we see that glycolysis, right, can produce at least 2 ATP, 2 net ATP per glucose molecule. As long as glycolysis can continue to occur, I can still produce ATP, right? So in order for ATP to be produced, that means this step has to occur. So in order for this step to occur, that means I must have NAD+. So therefore, the cell will go into anaerobic pathway, which the main purpose is to regenerate back the NAD. Okay, so let us see how anaerobic pathway can regenerate back the NAD+ so that the third stage of the glycolysis can happen, so that ATP can be synthesized without the presence of oxygen. Okay, so let's look at it. So when there is no final or terminal proton and electron acceptor, okay, there'll be no water formed, no electron being transferred along the ETC. So no protons will be pumped across the mitochondrial inner membrane, no proton gradient, no ATP can be synthesized from ADP and PI. So NADH and FADH cannot be oxidized. So NAD plus and FAD plus cannot be regenerated. Okay, so all you clear in this part now. So now Krebs cycle and link reaction will stop. But what about glycolysis? Okay. So let's look at glycolysis, how glycolysis can help to regenerate NAD+. So here we start with 1 glucose, undergo glycolysis to form 2 pyruvate molecules. And these 2 pyruvate molecules, without the presence of oxygen, it will go into anaerobic pathway. So pyruvate is converted to lactate. 2 pyruvate, 2 lactate. So you can see that 3 carbon and 3 carbon. So both are 3 carbons. So the only changes is the hydrogen atoms from the NAD, from the reduced NAD is being released to the pyruvate. So which means to say that pyruvate being the intermediate product of respiration. So pyruvate is an intermediate product of respiration. It acts as the alternative hydrogen acceptor because now oxygen cannot accept the hydrogen from NADH, right? So, but pyruvate go and accept the hydrogen from the NADH. That means pyruvate now act as an alternative hydrogen acceptor, alternative to oxygen. Okay, so now pyruvate being intermediate product of respiration act as an alternative hydrogen acceptor. So it accepts the hydrogen from the NADH and NADH, and H plus, okay, so that NAD plus can be regenerated. So when NAD plus can be regenerated, so let's write here, regenerated, okay, put here. Okay, So therefore, glycolysis can continue. So when glycolysis can continue, that means ATP can continue to be produced by substrate level phosphorylation. Okay, so now you can see that it's wonderful. The anaerobic pathway is very wonderful because it can make use of whatever resources that it has here. Whatever resources that it has here, okay, which is the pyruvate, and make the pyruvate receive the hydrogen first. Can you please accept hydrogen for me, please? I'm urgent. So pyruvate said, yes, I accept it. Okay, so you can produce back the NAD+. So it's a wonderful process. And this process can occur in our cell. So it's a very wonderful programming. Okay, can you see that the design of living organisms are really, really wonderful one. It's amazing, it's a miracle. And miracle is happening every day in our body. Okay, so remember that every day in your body, miracle is happening. So we are very, 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 very blessed. The fact that we are alive here, right? We are actually blessed every single day. Okay. Are we blessed every single day? Joyce?
Yes, teacher.
You feel blessed every day?
Yeah.
Very good. Lim Yi, how do you feel blessed every day? Or at least now lah.
Yeah.
You do, okay, very good. Okay, so every day you wake up, you can see the sunlight, right? That means we are blessed because the sun energy that comes to earth, and then we've got plants to produce food for us, and then we've got food to eat. Okay, so we are blessed all the time. We're always supported. It's always enough, more than enough, whatever that you need. You need, not you want, you need. Okay, right. So let's continue to, okay, so, right, so pyruvate receiving hydrogen, right, will become reduced. So this process is called reduction of the pyruvate as well as regeneration of NAD. Okay, now what is the name of the enzyme? You need to remember the enzyme that catalyzes this reaction is called lactate dehydrogenase. Okay, lactate dehydrogenase. Now some of you might actually ask me, do you see that? Teacher, if you say lactate dehydrogenase is removal of hydrogen from lactate, right? If I remove hydrogen from lactate, is my reaction moving to the left or moving to the right? This is to the left. This is to the right. If you say, teacher, lactate dehydrogenase enzyme catalyze what? Dehydrogenation of lactate, you know. So dehydrogenation of lactate is moving to the left or moving to the right. Please type your answer. Left or right? Yeah, I disconnected already. Why do I want to switch in progress? Join. Sorry, I cannot see your typing now because my this one gone already. Okay, let me see. Yes, move to the right. Andrea and Lau Huyen say. Okay, you move to the right. So you move to the right. Why the name of this enzyme lactate dehydrogenase? Something wrong somewhere, right? Do you notice that? Okay. Now I'm going to tell you because there are two different lactate dehydrogenase. One lactate dehydrogenase catalyzes the reaction to the left side. Another lactate dehydrogenase catalyzes the reaction to the right side. So that's why I think they name it differently by lactate dehydrogenase 1, lactate dehydrogenase 2. 1 and 2 are different things, okay? Why? Because the substrate that's supposed to sit in the enzyme is different, right? Enzyme has got their specific active site shape, you know. So because one of them, the substrate is this one, right? The other one, the substrate is actually this one. So the active site shape must be different. So that's why it's totally different enzyme, even though the name's the same. So how to make it different? You say Michael, then one is Michael Yee. The other one is Michael Jackson. Then you know they're different. Even though they're Michael, they are not the same Michael. Okay, Right. So this is for mammals, right? One step is already enough to recycle or regenerate our NAD+. Okay, so this is just look at number of carbon. 6 carbon becomes 2 3-carbon compound becomes 2 3-carbon compound. Right. So 2 pyruvate per glucose becomes 2 lactate plus 2 ATP per glucose. So this is a very small yield of ATP per glucose compared to aerobic respiration gives us 38 ATP per glucose. Can you see that versus 38, right? Okay. So aerobic respiration is 19 times more efficient than anaerobic respiration. Okay. So this is anaerobic respiration in plants or fungi or yeast or microbes. So it's called alcohol fermentation or ethanol fermentation. So for them, the anaerobic pathway involves 2 steps instead of 1 step. So how you can remember this is very easy. Mammals are considered as higher organism compared to plant, fungus, yeast, and microbe. Okay, higher, more complex. So when it's higher, then we can imagine it as an adult versus a child. When an adult takes 1 step, the child will have to take 2 steps. Okay, or you walk with somebody very tall. The person only take 1 step, but they have to take 2 steps. So when he walk, you have to run. Okay, so when he walk, you have to run. So that's what happened. So for plant, fungus, yeast, and microbe, they have to carry out 2 steps process. So glucose to pyruvate is glycolysis. All right. So from pyruvate to ethanol, can you see CO2 is being removed? So this process, what is the name for this process? Sufyan?
Decarboxylation.
Fantastic. Yes, this is called decarboxylation. Okay, so pyruvate having 3 carbon. When you remove a carbon, how many carbon is ethanol? You have learned in chemistry that it's 2, isn't it? So it's 2 carbon. So this process is decarboxylation. Okay, so after that, ethanol will be converted to ethanol by receiving the hydrogen from the NADH. Okay, so ethanol will be reduced to ethanol, and then NAD plus will be regenerated. So it is in the last step of the reaction that the NAD plus is regenerated. So the substrate of the last step will act as the hydrogen acceptor. So in this case here, what is the organism? What is the molecule that will accept the hydrogen? Charmaine? What is the molecule that will accept the hydrogen? Charmaine? Correct. So yes, correct. Ethanol is the one that will accept the hydrogen. So therefore, in this case here, ethanol, hello, where's my ethanol? Okay, in this case here, ethanol will act as the hydrogen acceptor. So it will act as an alternative hydrogen atoms acceptor, alternative to oxygen, and it is an intermediate product of respiration. Okay, so therefore you can see that in anaerobic respiration, it is the intermediate product of respiration that will act as an alternative hydrogen acceptor in order to regenerate NAD+, so that now at least the last step of glycolysis can happen, so that at least ATP can be synthesized by substrate level phosphorylation in glycolysis. Okay, so the equation will be 1 glucose gives 2 pyruvate and then converted to 2 ethanol plus 2 CO2 plus 2 ATP per glucose molecule. Okay, so this is again a very small yield of ATP compared to aerobic respiration. And the enzyme that catalyzes this reaction is called alcohol dehydrogenase or ethanol, because the alcohol here is ethanol. So it's ethanol dehydrogenase also. Okay, so here, what is the purpose of anaerobic respiration now? So let's just recap. Now you can see the big picture why already, right? So the purpose of anaerobic respiration is to use the intermediate product of respiration from glycolysis as an alternative hydrogen acceptor when there's no oxygen, so that NAD can be regenerated, so that glycolysis can occur, so that at least 2 ATP can be synthesized per glucose molecule when there is no oxygen. So this is a very wonderful design. Can you imagine that? And every single step needs enzyme. And who designed the code for the enzyme? So it's, oh, I mean, life is so amazing, right? Can you imagine that? Okay, so anaerobic respiration is less efficient than aerobic respiration. So now you can see why is it so much less efficient than aerobic respiration. So how are we going to explain in words? Tell me, why is it anaerobic respiration is less efficient? So there are a lot of reasons. The first thing is about ATP, okay? Less ATP, then give me the detail. How less is less? Sarah, how less is less? Zoom recording, okay. Now what I asked you to answer fast is, so 2 ATP versus 38 ATP per glucose molecule. So 19 times difference, okay, 19 times difference. So either 19 times more for aerobic or 19 times less for anaerobic, okay, less ATP. So other than ATP, what else? The product form from anaerobic respiration, like ethanol and lactate, they are toxic to the cell, okay? High concentration of ethanol can kill the cell, no? And then, why? Okay, very interesting. The membrane is made of phospholipid. Lipid can dissolve in ethanol and bye-bye. You know, the lipid, all the organelles surrounded by lipid, because lipid dissolve in ethanol and ethanol dissolve in water. Can you see all your phospholipid bilayer, the molecules, right? Already all surrounded by ethanol, surrounded by water, all dismantled already. No more membrane. Of course, the cell will die lah. No more membrane, no more mitochondria, no more nucleus, right? So the cell will die, definitely. It's like all the parts of the body dismantled, you know. Okay, so die, bye-bye. Okay, what about lactate? High lactate concentration. Lactate, right, is actually lactic acid. So when more and more lactic acid is produced by anaerobic respiration is when, therefore, when the acid increases, right? That means to say that your pH of the cell is going to drop. Okay, so when the pH of cells drop, that means to say that enzyme cannot function because our optimum pH is not so acidic, but now because of a lot of acid, so the enzyme cannot function. So and then a lot of processes like muscle contraction cannot happen. So you feel tired, so fatigue and muscle cramp. That means muscle cannot contract, jam up already. Okay, because enzymes cannot function to produce ATP, so muscle also jam up. Okay, so interesting, so they are toxic to the cell, right? Okay, third inefficiency is incomplete oxidation. Now this ethanol and lactate, are they organic molecule or inorganic molecule? Lao Huyen, Megan. Organic or inorganic molecule?
Organic.
Organic, so got energy or not?
Yes.
Can still be oxidized or not?
I think so.
Yes. An organic molecule fully oxidized becomes inorganic, right? Like CO2 and water, right? So since it's ethanol and lactate, it is still organic, it can still be oxidized. That means it's incomplete oxidation. These two molecules are still carrying energy. Thank you, Megan, for answering the question. So in plants, ethanol is actually excreted because it's toxic. So with a molecule that is having energy and the plant excrete it, is a waste, isn't it? Because the energy is not being harnessed and used by the cell. So it's a wastage of the chemical potential energy, right? In animals, we don't waste it, because animal is higher, more complex. They have some internal design in it to reuse it back. Recycle, recycling of waste, okay? So in animals, lactate will temporarily store the hydrogen first because there's no oxygen. So lactate will temporarily be holding the hydrogen first, okay? And this create oxygen debt, which I will actually explain later on. We come back to this later on. So it will temporarily store the hydrogen. Lactate will not be thrown away by the cell. When oxygen is available, lactate in the muscle will be carried by the bloodstream to the liver in order to be converted back to pyruvate in the presence of oxygen in the Cori cycle. So this diagram here shows you the Cori cycle. On the right side is the muscle, left side is the liver. So this right side process is the anaerobic respiration that produces lactate. So this lactate in the muscle has been transported by the bloodstream to the liver, where whenever oxygen is present, lactate can be converted back to pyruvate again, the original state of the molecule before lactate is formed. Then from pyruvate, 20% can be fully oxidized to CO2 and water and ATP. 80% it can be converted back to glucose, transported by bloodstream back to the muscle to be used by muscles again. So which means to say the energy, the chemical potential energy in the lactate will not be wasted. It's only used to temporarily store the hydrogen when there's not enough oxygen. That means you created an oxygen debt. That means to say, I'm going to pay you back. Whenever oxygen is available, I'm going to pay you back so that you be converted back to pyruvate. So there'll be an oxygen debt that has to be paid by the cell when it produces lactate. Okay, so let us look at more about the oxygen debt thing. Okay. So anaerobic respiration is used to buy the time. Buy what time? It's like, for example, now you don't have cash, right? You go to buy zap fan, you don't have cash. Then you say, okay, never mind. I pay you with e-wallet. Last time, no, never mind, I pay you with credit card. Okay, so I pay you with credit card, and at the end of the month, the bank is going to pay you. So then you say, okay, now I don't have cash. I pay you with credit card first. You accept first. And at the end of the month, then you will get your cash. So now you use anaerobic respiration to tell the cell that, okay, now I don't have oxygen. Okay, I pay you with something. I pay you with something. I pay you with pyruvate. I pay you with ethanol. Okay. Then can you please produce ATP for me? So this is to buy time that you want ATP now. I want ATP now, but I pay you with something. I don't have oxygen. Okay, I pay you an alternative hydrogen acceptor. Please accept it first. Okay, when I got oxygen, I pay you back. This only mammals can do. The rest of the organism, right? Ethanol fermentation cannot do. Okay, so what will happen in mammals is like, for example, us. So this is a graph that shows you y-axis the oxygen uptake of the organism in volume per minute, and x-axis is the time in minute. So you see here at the beginning of the graph, okay, it shows you that the oxygen uptake is very low and consistently horizontal. So this is the amount of oxygen that's needed by our body to maintain the basal metabolic rate. Basal stands for basic. Okay, the very basic. So what do you mean by basal metabolic rate? It's like the metabolic rate at rest. At rest means when you are, for example, sitting down doing nothing, or you're sleeping, especially when you're sleeping. When you're sleeping, you're still alive, right? And you're still breathing, right? Why you're still breathing? Because you need to be alive. What happened? When you are resting, all the cells in our body are still alive, especially our heart must continue to pump. Okay, our heart will continue to pump, it's still working 24 hours, and our lung will have to still continue to ventilate. So the muscles involved in breathing are the diaphragm and the intercostal muscle of the rib cage. So even we are resting, there are a lot of cells in our body that are still very active, especially the muscle cells. So therefore, you will still need ATP. So whenever you need ATP, okay, so we start with cardiac muscle, cardiac muscles of the heart, and other muscles are still working, okay, contracting. So therefore, you still need ATP. So therefore, you still need to carry out aerobic respiration. So therefore, you still need oxygen. So that's why you still need oxygen. So this is the basal oxygen level needed for basal metabolic rate. So then you start to exercise. At this time here, you start to exercise. So when you start to exercise, meaning to say that the amount of work done by your cardiac muscle will increase, because now not only your heart pumping, your skeletal muscle starts to contract. So you need more ATP, which means to say that you need more aerobic respiration to produce the ATP, and you need more oxygen for the aerobic respiration. So immediately when your muscles are contracting already, working very hard, you also need a lot of oxygen to produce this amount of ATP needed for the muscle to contract. So therefore, your body will have to adjust in order to take in sufficient oxygen. So therefore, you can see that the oxygen level starts to rise from here up to here. And then when it level off, this is actually the actual volume of oxygen needed for aerobic respiration to produce the amount of ATP that's needed to maintain the work nature of this muscle. Some of you will walk slowly, some of you will run very fast, right? Some of you go for competition. So the amount of work done by the muscle is different. So the higher the work done, that means the more oxygen you need lah. So it depending on the work, therefore the oxygen needed. So this graph here shows you this is the amount of oxygen needed. So when you start to run, right, at that rate, you need this amount of oxygen, but your body cannot immediately provide the muscle with this amount of oxygen, because your body needs to adjust in order to be able to get this amount of oxygen. So what kind of adjustment here is, how do you get oxygen into your muscle? You must just get it from the air, right? So you need to adjust loading. The loading rate of oxygen must increase, and then the transport rate of the oxygen must increase, as well as the unloading rate of the oxygen must increase. Then only your muscle can get the volume of oxygen needed. So all these three things must adjust: the loading, the transport, and unloading of oxygen. The rate must increase. So loading rate. So where do we load oxygen into our body? From where? Kirti? From where do we load the oxygen into our body?
Red blood cells.
From? Hello? It's not red blood cell? Not red blood cell. Hello, where do you get the oxygen from? From your red blood cell?
No,
Into your body?
from the lungs.
Where?
From
the lungs, yes. First, oxygen is outside our body. In order to bring it in, we must start with the lung first. So it's from the lung, right? Okay. Then transport the oxygen in which part of our body? Samir? What will transport the oxygen throughout the body?
Red blood cell.
Yes, red blood cell means in a red blood cell in the bloodstream, right? So in short, the blood. Okay, the transport medium. Then unloading. Unloading will occur in the muscle. That one no need to say it. Okay. So there are three types of adjustment that's needed in order to increase the oxygen needed by your body. So in order to increase oxygen loaded by your lung, what will your lung do? Or how will your lung adapt? Tan Jiaying? What will your lung do to take in more oxygen?
Inhale.
That means to say you don't exercise, you don't inhale lah. How is your inhalation different when you're exercising compared when you're sleeping?
Take more oxygen inside.
How do you take in more oxygen?
Red blood cell take in more.
No, lung, your ventilation rate will increase. Your ventilation rate will increase, meaning to say that your breathing rate will increase, plus breathing depth will increase, right? You breathe in deeper and you breathe in faster, right? So this is the adjustment for your lung. Ventilation rate increases. to take in more oxygen. Now, for transportation of oxygen in the blood, how do you increase the transportation? Ryan Kwa.
Increase the pressure.
Increase the what?
Pressure.
Pressure, very good. So increase pressure means what?
It flows faster.
Stroke volume. What you mentioned there is actually stroke volume. Okay, so increasing stroke volume is how much volume of blood being pumped out per stroke. So stroke volume plus heart rate is equal to cardiac output. Please write all this into your notes. Okay, so we are going to increase cardiac output. Cardiac output means how much volume being released per unit time. I cannot write this per unit time, -1. Okay. Yeah. Per unit time, put time at that. Okay. So cardiac output is equal to stroke volume times heart rate, because stroke volume is how much volume per stroke. Whereas heart rate is how much volume per beat, and heart rate is how many beats per time. Right, so you put both together, you cancel off, then it will give you beat and beat cancel off, it give you volume per time, right, which is volume per time, cardiac cycle. So both breathing rate, ventilation will increase, and also cardiac output will increase. So that will increase loading rate and transportation rate. Now, what about unloading rate? Do you remember there's a process whereby when CO2 concentration increases, your hemoglobin will unload oxygen. What is the name of that process? Joyce?
Sorry, teacher, can you repeat?
When CO2, when partial pressure of CO2 in a tissue increases, it will cause hemoglobin to unload oxygen. What is the name of this process?
More shift.
Yes, very good. Fantastic. Okay, give her a clap. Okay, Bohr effect. Bohr effect. So Bohr effect says that whenever there's a high partial pressure of CO2, this will cause the oxygen dissociation curve to shift to the right side. Right side means reject, right? So why does it shift to the right side? Because you know that the CO2, right? You know the process of CO2 being transported by the hemoglobin, right? CO2 in a hemoglobin will be converted to H2, converted to H2CO3, right? Which will dissociate into H plus what is this? And HCO3 negative. Right? Okay. And this H plus, now hemoglobin is carrying oxygen towards the tissue. So this hemoglobin is holding the oxygen, right? It's actually 8 HbO8. Never mind, I just use HbO2 to represent 1 O2. So whenever the acid is present, remember I already said before, hemoglobin has got a higher affinity for H plus than oxygen. So given a choice when oxygen is present and H plus is present, it will turn and bind to H plus rather than oxygen. That's why oxygen is being released. So that's why whenever CO2, partial pressure CO2 is high, the oxygen dissociation curve will shift to the right side because hemoglobin will actually release the oxygen. It will have got lower affinity for oxygen because it has got new partner, H plus. So therefore, it's got higher affinity for the new one. Okay, right. So can you see that? So this is how our body have to adjust before it can bring in the amount of oxygen or the volume of oxygen that's needed by the muscle cells in order to produce enough ATP. So which means to say, during this time period here, okay, some of the cells will have to go into anaerobic respiration to produce the ATP, right? Because we need this amount of oxygen here. We need this amount, but the oxygen level does not reach this amount yet. So for this amount that is insufficient, the cell will have to undergo anaerobic respiration to produce the ATP. At this time here, this amount of oxygen is insufficient, then it has to go into anaerobic respiration to produce the ATP. Until here are then sufficient oxygen already, then no need to carry out anaerobic respiration anymore, or aerobic. So that's why those of you who exercise a lot, you can actually feel for yourself that at the beginning of the exercise, when you start jogging or swimming or whatever, right, is like quite difficult. Then after some time, you find that, hey, it's like practically you're on auto cruise mode. You know, auto cruise is that you don't have to put in energy, it runs by itself. You know what I mean? It's like you don't have to pump in extra energy. Okay, it's just run by itself only. So that's when you achieve this state already, right? All aerobic already. Okay, so it's wonderful. Huh? So now during this stage here, the blue color region here, right? When the cell carry out anaerobic respiration because of oxygen deficit, it will undergo anaerobic respiration to generate the ATP, the amount of ATP that's required. So during that time, they will produce lactate, right? They say, I'll pay you first lah, I need ATP. So if you pay the cell first with something else, you have to pay back with oxygen. So when you pay back? You pay back when you go to rest. So when a person stops exercising here, right? So you're resting already from this time onwards. So this is called resting already. Okay, resting. So when you're resting already, that means you stop exercising, your skeletal muscles are no longer contracting anymore. Okay, so during that time, right, you look at this green color area. During that time, you're already resting, but the oxygen level taken in by the body is still higher than the basal metabolic rate here. Can you see that? Oxygen level that's taken in is still very high. It will take some time for it to slowly decrease until the basal metabolic rate level. So there will be excess oxygen being taken in even though skeletal muscles are resting. So this region is called the oxygen depth. So this is the amount of oxygen that's needed to pay this oxygen deficit. That means you're using this oxygen that's taken in here now to convert the lactate back to pyruvate, to pay back whatever debt that you have incurred at this time. Okay. Now, the next thing I want you to see is that, can you see the green color area is bigger than the blue color area? So you tell me, teacher, how come I owe you 10 ringgit, right? After I finish exercise, I have to pay you back 15 ringgit. Can you see this area is bigger? Now, it's not 15 ringgit, you also pay back 10 ringgit. The extra 5 ringgit, right, is because the moment you stop exercising, your heart rate, your breathing rate will not suddenly like drop from, let's say your heart rate will not suddenly drop from 140 all the way to 60. Yeah? Wow, suddenly stop exercising, suddenly heart rate goes back to 60. No, right? It takes some time for slowly decrease. 150, 40, 30, okay, 90, 80, 70. Some of you 80 something, some of you 70, some of you 60 something. Normal. So that means even though you stop exercising, your heart, okay, is still pumping very fast, your breathing rate is still very fast. So therefore, there will be excess oxygen taken in to maintain the higher rate of work done by your heart as well as your lung. So that's why there's excess oxygen here. So this oxygen here is in total more than the one over here. Okay, so that is the concept of oxygen debt that we need to pay whenever we go to anaerobic respiration. So when oxygen is available, we have to pay it back, means convert lactate back to pyruvate, so that can be used for aerobic respiration. So today we have looked at anaerobic respiration. In mammals, pyruvate is used as the alternative hydrogen acceptor to produce lactate. In yeast, ethanol will be used as an alternative hydrogen acceptor to form ethanol. Okay, function is of the anaerobic respiration is to regenerate NAD so that at least 2 ATP can be synthesized per glucose molecules whenever there's no oxygen. There will be inefficiency in anaerobic respiration in terms of only 2 ATP produced. The products are toxic and also incomplete oxidation, so energy can be lost. But mammals got Cori cycle to convert the lactate back to pyruvate whenever there's enough oxygen. So anaerobic respiration is for us is to buy time, and that creates oxygen debt in mammals only. Okay, so that's all for today's lesson. So we've looked at the entire respiration in terms of aerobic pathways, which involve glycolysis, link reaction, Krebs cycle phosphorylation, and anaerobic pathways, which involve glycolysis and the anaerobic pathway. So that's all for today's lesson.